Hello, everyone. Thank you for joining the Business of Biotech at the Moffitt Cancer Center. My name is Sungjune Kim. I'm an associate member in the Department of Radiation Oncology and also with a secondary appointment in the Department of Immunology. I'm a physician scientist, and our lab is focused on epigenetic immune regulation. With this particular project, we've been working on methylation of the tumor cells and how that affects the tumor immunity. So to quickly go over, we have long known there is an interaction, we call it immune synapse, between the antigen presenting cells and the tumor cells and the T cells. The interaction has been mostly described between the antigen presenting cells, mostly dendritic cells and T cells, where most of the classical concepts in immunology has arisen. More recently, we have found that the tumor cells are also expressing all these immune-related proteins to modify the T cell behavior. And the question we asked was, how does methylation potentially impact this interaction? Very interestingly, when we looked at the methylation status of those immune-related molecules, all these molecules, for instance, here, as you can see, CD40, which is on the stimulating side for the immune interaction, is hypermethylated in tumor cells across different tumor sites. On the other hand, the inhibitory side was hypomethylated. And in terms of the role of methylation in protein expression, it's actually opposite. So when we have hypermethylation of this particular gene, their expression is suppressed, as you can see here. And opposite is true for these inhibitory genes. So that phenotype was quite interesting. We did further bioinformatics study using principal component analyses, and it has shown that the tumor cells either really hypermethylate the stimulatory genes or hypomethylate the inhibitory genes as compared to these red tissue types which are normal adjacent tissue. Not only that, this particular principal components were prognostic. Here I'm showcasing melanoma survival, where when you have high principal component 1 score, they tend to have worse survival. And these are the patients who have hypermethylated stimulatory genes. What was more interesting, and this data is still preliminary at this time round, we looked to see if this principal component is also associated with response to immunotherapy. And it makes sense because the whole biomarker that we developed is based on the immune proteins. And as you can clearly see here is that the patients with high PC1 score, principal component 1 score with hypermethylated stimulatory genes had a worse response to immunotherapy. And this worsened immunotherapy is translated into worse outcome, survival outcome as well. So these studies are currently ongoing to really dissect the mechanistic background and also the clinical utility for application to immunogenic tumors like melanoma, lung cancer, head and neck cancer, and renal cell carcinoma. And this is a nice summary of the working hypothesis that we are testing, where the stimulatory gene hypermethylation and inhibitory gene hypomethylation leads to altered interaction with the T cells, leading to differences in cytokines, immune cytokines, and immune effector molecules to affect the tumor immunogenicity. Thank you for listening, and I'll be very happy to discuss further as needed.